Well, thank you, Claude. It's a real pleasure to be here. It's the first time I've been here, and it's really a beautiful place. So thank you for the invitation. Yes, Claude and I know each other quite well, going back almost 40 years now. It's hard to believe, and that's probably why he felt he had to remind me at least 6 times that you are a mixed audience of experts and experts and non-experts, and I should therefore tailor my talk accordingly. Of course, this had the effect of making me very nervous because it's a very difficult thing to do. I mean, if I make the talk too technical, the non-experts will get irritated, and if I don't make it technical enough, the experts will get irritated. So it's a no-win situation. I know I'm going to come out here being hated by everybody. So what I decided to do was to mix a little bit of the two, have some material that is not very technical, and then I'll have some which is technical. So there will be something for everybody, although not everything will be for everybody. So in the end, I hope I leave with at least a few friends and not a room full of enemies. Okay, so what I'd like to do today is tell you about mTOR. That's what we work on, and cell growth. And I would like to tell you about a recent story from the lab, one which we've not published. It's a story about which I am extremely excited. I haven't been this excited about something in a long, long time. Probably I've never been so excited about something coming out of the lab. But before I do that, and that's about this subject of how diet can cause diabetes. But before I do that, I would like to give you an introduction to mTOR signaling, or mTOR signaling. I would like to make this a sort of easy listening kind of introduction, so it'll be largely historical. And then we'll eventually get into the perhaps the more technical part of the talk. Okay, so this is a condensed version of the history of the mTOR pathway, at least up to about 5 or 10 years ago. This here is the very first ever model of mTOR signaling published. It's one we published shortly after our discovery of mTOR in 1991. I have to say I'm actually embarrassed I published this model. Who here remembers Gary Larson? Anybody? Okay, a few. He was a very famous cartoonist, long retired now, I suppose, a scientific cartoonist. And that's what this actually reminds me of, is a Gary Larson cartoon, more than an actual serious scientific figure from a published manuscript. It shows mTOR exists, and then question marks everywhere else. So it basically says mTOR exists and nothing else. So, of course, we were much younger in 1991. The picture today is considerably more complex, and what's driven this increase in complexity has been this exponential increase in publications in this area. And this, in turn, has been driven by the fact that mTOR turned out to be both fundamentally important and clinically important, much more so than we anticipated when we first came across it back in 1991. So what I'd like to do with the introduction is take you back to 1991 and weave our way through the years until we get to something resembling what the mTOR pathway is. So I'll layer on the complexity more or less as we went through it through the years. So here's the timeline. The story actually started much earlier, in 1991. It started in 1965. And that's when a group of scientists from Montreal went to Easter Island, Rapa Nui, and they were prospecting for exotic microbes which they hoped would produce novel metabolites which they could then develop into an antifungal. They did isolate such an organism, such a bacterium. They did indeed isolate a novel metabolite from it, which they called rapamycin, after the island of Rapa Nui. And it did indeed have antifungal activity, which they started to develop for clinical use. Now, the problem was that as they were developing it, they found that it had the very undesirable side effect of suppressing the immune system. And of course, suppressing the immune system is the very last thing you want to do to somebody who has a fungal infection, so it was immediately dropped. Now, During the 80s and 90s, immunosuppressive therapy, not modern day immunotherapy, but immunosuppressive therapy came into existence in the clinic for transplantation patients. So if you'd had a liver transplant, for example, in 1980, the chances were 80% that you would not survive. Today, if you have a liver transplant, the chances are 80% that you'll survive. And this is due to immunosuppression as a therapy. It is now a viable therapy used in the clinic. And it was developed by use of drugs such as rapamycin, there are others, cyclosporin A and FK506, but it was these drugs which then exploiting this immunosuppressive activity which led to this immunosuppressive therapy. So it was repurposed, rapamycin was rediscovered and repurposed and applied for the very reason which it had really been rejected as an antifungal, to suppress the immune system. So this is a major development in medicine, and if you go to Rapa Nui, or Easter Island, you actually find a plaque commemorating the place where these scientists picked up this handful of dirt which contained this bacterium which produces rapamycin. And this is written in Portuguese for reasons I don't quite understand, but it says that in this place was collected in '65 a sample that allowed not only the isolation of rapamycin, but also ushered in a new era in transplantation surgery. So this commemorates the fact that it became a immunosuppressive drug. I actually always wanted to go to Rapa Nui. It's my kind of mecca, given my long career in working with rapamycin. And happily, I actually managed to do that just a couple months ago for the very first time. So we took a picture. The first thing we did after we dropped our bags to the hotel was to go directly to the site on the island where this monument is. And we got there, and lo and behold, someone had stolen the plaque. But the site was still there, so we were very happy at least to see where the plaque had been. This is a group of scientists. This is Nahum Sonnenberg. Some of you might know Nahum from this field. So there was a bit of disappointment there, but we, of course, enjoyed our trip to Rapa Nui nonetheless. This is what rapamycin actually looks like, this natural molecule secreted by a soil bacterium. It's now found use in 3 major therapeutic areas, not only transplantation, where it first became famous. It's now even more famous as an anti-cancer drug, based on its ability to block cell growth, as I'll come to later. And it's even used in the clinic for treatment of cardiovascular disease, restenosis after angioplasty in particular. So this molecule had a very humble beginning in the soil of this very isolated place, has had major impact in the clinic. And the fact that it can be used to treat in 3 so very different therapeutic areas suggests that whatever the target is, it must be something rather central to the cell. And this is what led us to be so excited about this molecule in the late 80s. It was known it worked in all these contexts, but the mechanism of action of the drug was unknown. And this is what we wanted to study. An outstanding postdoc joined the lab at the time by the name of Joe Heitman, who had a medical background and also was very interested in trying to understand how this drug actually worked. So what Joe decided to do, which was rather unusual, was to take a genetic approach with yeast to understand how this drug, which is being developed for yeast in humans, worked. So people thought, and I'll blame Joe, thought Joe was rather unusual for giving this human drug to yeast cells, like giving aspirin to a yeast cell. Why would you ever want to do that? It was viewed as completely physiologically irrelevant. But of course, Joe was making the assumption that whatever the molecular mechanism of the drug, it would be conserved all the way from yeast to mammals, and therefore you could use yeast, and particularly yeast genetics, to elucidate the mechanism of action of the drug. And what Joe, of course, knew, and what most others had forgotten, was that rapamycin had originally been isolated as an antifungal, and therefore would have activity on yeast cells, which is a fungus. So Joe decided to apply the drug or the molecule for what it was evolved to do. And from that perspective, you could say that giving this molecule to humans is the strange idea, not giving it to yeast cells. So Joe isolated mutants, yeast mutants, as his approach to understand the mechanism of action of rapamycin, in particular the target in the cell of rapamycin. And we did that. He isolated mutants which were defective in any one of 3 different genes. Most of the mutations were in this gene, which he called FPR1. This is a gene he'd already characterized earlier in a different project. And these mutations are only common, but they're recessive. And then he also isolated mutations in 2 new genes, which he called TOR1 and TOR2 for target of rapamycin. Of course, he didn't know this was actually the target, but because these are just, at this point, just mutations or genetic loci in the yeast genome. These mutations were not only rare compared to these mutations were also dominant. So why do we get mutations in 3 genes, or why did Joe get mutations in 3 genes, some which were common and recessive and some were rare and dominant? Well, the answer to that question relates to the mechanism of action of the drug, and this is how the drug works. So this is rapamycin, now drawn in a different configuration, a space-filling configuration. And this protein is the FKBP protein, which is the product of that FPR1 gene. So the toxic agent in the cell is not rapamycin by itself, but rapamycin in complex with FKBP. Rapamycin alone does nothing. So, and why you need the complex to get toxic activity is something I'll get to later. In any event, since FKBP, this small protein, 12 kilodalton protein, is absolutely essential for drug activity, although not essential for viability of the cell, any simple loss of function mutation anywhere in the gene encoding this protein will give you a complete rapamycin resistance, and these will be, of course, recessive mutations. So this is why most of the mutations were in the gene encoding FKBP, in the FPR1 gene. TOR, on the other hand, it's a very large protein. It's about 300 kilodaltons in size. So this is a very small part of the TOR protein. If we had the whole thing here, it would fill a good part of this stage. In this case, TOR is absolutely essential for cell viability, and therefore it can tolerate very little mutation. And in fact, every mutation we obtained in the TOR genes fell into a specific codon which determined a key residue in this alpha helix, which was a contact site between TOR and rapamycin. And the effect of these mutations was obviously to prevent the binding of FKBP rapamycin to TOR without otherwise affecting TOR activity. And this is why these mutations are very rare. They are confined to a single codon, and why they are dominant is because they naturally confer rapamycin resistance even in the presence of a wild-type copy of the TOR gene. So this experiment which Joe did, which is a very simple experiment, he simply made a plate of rapamycin and streaked some yeast cells on it, turned out to be extraordinarily informative because these mutations not only elucidated the mechanism of action of rapamycin, the fact that it has to form a complex with FKBP to be toxic, it also identified the FKBP rapamycin binding site in the TOR protein, but most importantly, it led to the identification of TOR itself, which we could eventually see once we'd cloned and sequenced the TOR genes. And needless to say, we used the mutations in the TOR genes to clone the TOR genes. So once we cloned and sequenced the TOR genes, the first thing we observed was that the 2 TOR genes encode very similar proteins. The proteins are 70% identical, and they also turned out to be the founding members of this class of atypical protein kinases called PICs, for PI kinase-related protein kinase. So what all these kinases have in common, all these protein kinases, is they look like PI kinases or lipid kinases, but are in fact protein kinases. And to this day, we don't know why they all resemble lipid kinases, but are in fact protein kinases. No member of this class actually has lipid kinase activity, at least demonstrated to date. Since the early discovery of TOR in yeast, we now know TOR is found in all eukaryotes, all the way from yeast to human, including worms, flies, and plants. But yeast is unique in that it has 2 TOR genes and thus 2 TOR proteins. All other organisms have a single TOR gene, therefore a single TOR protein. The nomenclature system, which just introduced you to that, based on the original names from yeast, this name TOR has been kept through in all eukaryotes with a little prefix to indicate where it's from. mTOR originally meant mammalian TOR. Now it's been corrupted to mean mechanistic TOR. Some people still use mammalian TOR, though. So this is where we were in about the year, 1993. We knew TOR was a highly conserved protein, essential protein, most likely a kinase, but we didn't know what its cellular role was. What purpose did it serve in the cell other than to be the target of rapamycin? Of course, it didn't evolve to be the target of rapamycin. So a great deal of work from our lab and a number of other labs then started investigating what is TOR actually doing in the cell, and this is where we made this discovery that the role of TOR is to control cell growth or accumulation of cell mass, which eventually determines cell size. And this is most photogenically illustrated in experiments done in Drosophila, and this also underscores how highly conserved the TOR proteins are. You can study it in almost any organism. These are experiments done by 2 Drosophila geneticists, Tom Neufeld and Bruce Edgar, both in America. In this experiment, what Tom Neufeld did was to selectively inactivate TOR in these 2 GFP-expressing cells of the fat body of the fly. This is like the liver of the fly. And as you can see, these cells end up being smaller than their neighboring wild-type normal cells. So the obvious conclusion from this is that the role of TOR is to control cell size. Now, Bruce Edgar did the inverse experiment. He hyperactivated TOR signaling or TOR activity in these in the green cells. In this case, he found the cells became much larger than the neighboring wild-type cells. So the conclusion is TOR controls cell size, presumably by controlling cell growth. Now, Tom Neufeld took this experiment one step further. He started looking for TOR for mutant flies which had a whole body defect in growth control, so growth modifiers. And he isolated such a fly. It's important to point out this fly is what fly geneticists call a hypomorph. In other words, it's a partial loss of function. It's not a complete loss of function, because if it were a complete loss of function, this fly never would have been born. This would have been a lethal event very, very early in development. In this case, this is a mutation in the promoter of the TOR gene, which reduces TOR activity, but does not eliminate it. So when Tom Neufeld isolated this fly, this smaller but perfectly proportioned fly, he then asked the question, why is this fly smaller? Now, there are 2 possibilities. One, it has the normal number of cells, but each individual cell is smaller. Or it has the normal size of cells, but just less cells. So is this a problem of cell size or cell number? And how would you actually answer that question? How would you actually investigate that? Well, what Tom Neufeld did was he looked in the wing, which is a plane of cells. So it's very easy to count the number of cells and also to measure the circumference of cells. And from there, you then extrapolate to the whole fly. And what Tom found was that this fly has more or less the normal number of cells, but each individual cell is slightly smaller to give rise to this fly. So the conclusion from this is that TOR not only controls cell size, but thereby also controls tissue size, organ size, and ultimately size of the whole animal. To me, this is an absolutely astounding result, because to make a fly like this is a program in 4 dimensions. And here it seems this program seems to have been rewritten at least 3 of these dimensions by a single mutation in the genome, in this case a mutation in the promoter of the TOR gene. So you can think of all the regulatory circuits, all the metabolic circuits, which have to be rewired to create this smaller but perfectly proportioned fly. Now, it's also important to realize TOR controls growth in many other contexts, oftentimes independent of cell division, as would have been the case of, where in the case of this developing fly, it would have controlled growth in the context of dividing cells. But we have many contexts independent of cell division. For example, if you fast or feed, your liver changes in size, and this not because you acquire more cells or lose cells, the size of individual cells change. And this is controlled by TOR signaling. Very common in one of my favorite examples is muscle. You know, if you go to you exercise, your muscles get bigger, and that's not because you get more muscle cells, but the muscle cells you have get bigger. Interestingly, TOR has gone mainstream, breaking into popular culture in this context. If you go online, you'll find products like this, NTOR, which I suppose stands for anabolic TOR. This is a dietary supplement for weightlifters. And if you look very closely in the label, you'll see things like FOXO and PKB and mTOR. So I'm not sure bodybuilders know exactly what all these things are, but in any event, I don't even know what's in this flask. I tried to buy it. I couldn't buy it in Switzerland. You had to be in America. But my feeling is all that's in it is leucine for reasons I'll come to later. It gets much worse than that. I always wanted mind-blowing results in the lab. So here we have mTOR, which stimulates a powerful mTOR signaling activator. Again, I have no idea what's in here, but I certainly wouldn't buy it and use it. Anyway, back to more serious matters. In the central nervous system, there are very dramatic changes, not only in cell size, but in cell shape. And this has also been shown to be controlled by TOR. And finally, it's actually been shown that queen bees become larger because of the TOR pathways upregulating the larvae, which give rise to the queen bee, because they're fed a royal jelly, which is hypernutritious and upregulates the TOR signaling pathway. So the take-home message here is that TOR controls growth very broadly in terms of physiological context and broadly in terms of eukaryotic organism. Essentially, any case in eukaryotic biology where you see a change in cell size, there's a high likelihood that TOR signaling pathway is responsible for that. So how does TOR actually do this? How does TOR control cell growth and cell size? Well, again, work from our lab and a number of other labs have shown that TOR controls a large number of cellular processes which collectively determine mass accumulation and thereby cell size. And these processes can be subdivided into two groups: the anabolic processes which TOR activates and the catabolic processes which TOR inhibits. So TOR balances these opposing forces of synthesis and degradation such that the appropriate level of mass accumulates relative, and this is important, relative to the available nutrients. So in very simple terms, TOR is what makes us grow or controls our growth when we eat. So how does TOR control all these processes? Well, effector signaling pathways emanate from TOR to intersect with key proteins involved in all these processes. In some cases, we know what those pathways are. In some cases, we don't. But here's the picture where we were in the year 2000. And this is a yeast cell, and you can tell because we have two TOR proteins. And the picture that emerged here is that there are, in fact, two TOR signaling pathways. One is mediated by TOR2 alone, so we call this the TOR2 unique pathway. And this controlled organization of the actin cytoskeleton, which determines how the secretory pathway is oriented in the cell, and therefore determines where in the cell new mass is laid down. So we view this pathway as mediating spatial control of cell growth. This pathway over here, which is more complex, and this one, either TOR1 or TOR2, can serve in this pathway redundantly, activates all these processes which actually lead to upregulation of mass in the cell. And this is what responds to nutrients. So we view this branch of TOR signaling over here as mediating temporal control of cell growth. So we think the overall logic of this network, and we call it a network because it's more than a single pathway, we think the overall logic of these two branches to the network, or the two pathways to the network, is that they integrate temporal and spatial control of cell growth. And of course, these two aspects of cell growth have to be integrated for growth to occur at the right time in the right place. And what better way to do that than to have the TOR protein doing both. So this is where we were in the year 2000, but we had many, many questions. For example, what determined the specificity of the TORs? Why could only TOR2 signal here, but either TOR could signal through this branch? We knew FKBP rapamycin bound and inhibited TOR over here, but couldn't inhibit TOR over here, even though it would be TOR2 in both cases. So what determined the ability of rapamycin to inhibit only one branch but not the other? And to address these questions, we decided to take a biochemical approach. Everything it's shown here we did by taking a genetic approach, and we pushed that as far as we could, and then we decided to supplement our genetic approach with more biochemistry. And we decided to purify the TOR proteins out of yeast cells. And the idea was to use a very gentle purification scheme, so we'd co-purify any proteins that came with it, and therefore we'd get more insight into what proteins TOR actually interacted with, and then maybe this would lead to answers to these questions I just posed about the specificity of the two TORs and why only one can be inhibited by rapamycin. We did that. We isolated two complexes, which we isolated TOR, and we found that these isolated as two different complexes, which we called TORC1 and TORC2 for TOR complex 1, TOR complex 2. We had these two complexes isolated in a test tube. We could then sequence the proteins, and with this information, we could then isolate the genes. And once we had the genes, this all being yeast, we could knock out the genes very easily. And we did that. We found that these two complexes corresponded to these two previously identified branches of TOR signaling. So this was a very gratifying moment in the lab because of the convergence of about 10 years of genetic research identifying the two pathways and a few years of biochemical research which identified the two complexes into a unified model of TOR signaling, which we call sometimes the two-branch, two-complex model of TOR signaling. And of course, the biochemistry and the genetics cross-validated each other, so we had a very high level of confidence in this model. So this is where we were in the year 2002. We then wondered whether the two TOR complexes were conserved all the way from yeast to human, like TOR itself. So at that time, we looked in the genome databases, which were incomplete. There was no complete human genome sequence at that time. There was one incomplete, and when we looked at this one incomplete genome sequence, we found some proteins which had some similarity to the proteins which make up these two complexes in yeast. So this suggested that the two complexes might be conserved. We started doing experiments with mammalian cells, but also others, most importantly David Sabatini at MIT was also working with mammalian cells, and we all showed that also Kazuyo Nezawa in Japan and Kulian Guan in San Diego, we all showed that the two complexes are indeed conserved all the way from yeast to human. In fact, they phosphorylate the same proteins, or orthologs of the same proteins, also have similar proteins which are upstream. So the picture then emerged as that the TOR signaling network is a primordial or ancestral signaling network which has been conserved throughout eukaryotic evolution to control this very fundamental process of cell growth. Now that statement I just made is not entirely accurate because there's another part of the network up here which evolved much later. This is the insulin or growth factor signaling pathway, and this evolved much later. This evolved with multicellularity and was then grafted onto the more primordial TOR network which already exists in unicellular organisms. And the reason for that is that growth in multicellular organisms or metazoans is growth control in these organisms is more complex than growth in unicellular organisms, because in multicellular organisms, the growth of every cell in the body has to be coordinated with every other cell in the body, such that our organs end up being properly proportioned. So in metazoans, we need a input into the growth controlling TOR network to coordinate growth over a whole body plan. And that then is what the growth factor signaling pathway provides and why this evolved with multicellularity. So we now have, this is where we were in about the year 2005 to 2010. At about this time, also emerged that mTOR is involved, functionally linked to an unusually high number of diseases, all of which are characterized by inappropriate or ectopic cell growth. And this can be malignant forms of cell growth or benign forms of cell growth, but in all cases, inappropriate cell growth, which further supports or underscores this notion that mTOR is a so-called central controller of cell growth. More recently, mTOR has been implicated in another set of disorders, and this is what I'll come to later in the talk. It's been implicated in the metabolic disorders such as obesity and diabetes. We know that chronically high levels of nutrients. So I forgot to mention that in mammals, the mTOR is also activated by nutrients, amino acids in particular, and leucine is one of the most efficient amino acids in upregulating mTOR signaling, which is why there was probably leucine in that flask for bodybuilders. But in mammals, it's not only leucine, as I said, it's also a growth factor, and now we also know energy in the form of ATP via sensed via AMPK. So once these three factors are present, growth factor, nutrients, and cellular energy, the mTOR network is upregulated and growth ensues. So we know that chronically high circulating levels of nutrients can upregulate mTOR even in the absence of growth factors, and this can lead to adipogenesis and obesity, which in turn can lead to type 2 diabetes. But we had a more direct link to diabetes, a famous negative feedback loop in this pathway where mTOR phosphorylates IRS in the insulin signaling pathway to inhibit it. As a result, the cells become insulin resistant, which is one of the hallmarks of diabetes. This is one of the links between diet and diabetes, which I'll get to in more detail later. In this context, some have actually proposed that FKBP or rapamycin could be used as an anti-diabetic because if you inhibit mTOR, you inhibit the negative feedback loop and you restore signaling through the insulin pathway. I don't think anybody's actually developed rapamycin. This would be a fourth major therapeutic area for rapamycin. But I don't think anybody's actually developed rapamycin as an anti-diabetic because there are a number of complications which would prevent that. But something giving credence to this notion that it could possibly be done is that metformin, the world's most commonly prescribed anti-diabetic, works at least in part by downregulating mTOR signaling, much like rapamycin would. Okay, so another area in the mTOR field which has generated a great deal of excitement is the fact that it controls the aging process, so it controls lifespan. And this is exciting because it's been known since 1935, so that's 85 years ago, that dietary restriction or caloric restriction or simply eating less extends your lifespan. But the molecular basis of this was completely unknown until about 10 years ago. And that is shown here. It was shown here that rapamycin can extend lifespan. This was first shown in yeast, worms, and flies. In this paper, it was shown in mammals. And this is indeed the first pharmacological extension of lifespan in mammals. So we know that mTOR is a nutrient sensor. It responds to nutrients. And the effect of rapamycin is to block mTOR to trick the cell into thinking there are no nutrients there. So rapamycin treatment mimics the effect of eating less or dietary restriction. So this is why this has created so much excitement, particularly in the aging field, is that this now provides an explanation for how dietary restriction extends lifespan. If you eat less, the mTOR signaling network is downregulated. This leads to broad metabolic changes, the net effect of which is to extend lifespan. Okay, so this is where we were in about the year 2010. We had a fairly good idea of how mTOR controls growth of single cells. We had a very good idea about the physiological role of mTOR. But then there was what was, to my mind, an extremely exciting discovery made by another Drosophila geneticist by the name of Pierre Leopold, then in Nice, now in Paris. And what Pierre was doing was he was looking for flies which had a growth defect, much like that fly I showed you before, isolated by Tom Neufeld. And what Pierre isolated was a fly which was smaller, at least the larva of the fly was smaller, and the mutation was in an amino acid transporter which was specific to the fat body. So as a result of that, amino acids did not get into the cells of this one tissue, and as a result of that, that one tissue, the cells of that one tissue were smaller because TOR was not activated because amino acids could not get in. So the cells were smaller, but not only were the cells smaller, but now the whole tissue was smaller, the whole organ was smaller. So this made perfect sense. It fit exactly what we knew based on the state of knowledge at that time. But what he also observed, which was the really astounding thing for me, was that not only was the fat body small, but all the other tissues were also smaller, correspondingly smaller, so to match the size of the fat body, despite the fact that this amino acid transporter was not required by these other cells which had their own amino acid transporter, and therefore, in theory, mTOR signaling should be normal in all these other cells. So this told us that there's some mTOR-dependent signaling between tissues to control growth over a whole body plan, so our organs end up being properly proportioned. And this opened up a whole new horizon, a whole new level of growth control to investigate. And this is something we wanted to investigate further, not in Drosophila, but in our case, in mice. So the way to do this, and this is the work we started about 10 years ago, was to knock out mTORC1 or mTORC2 specifically, or alone, in different tissues. So we now, tissue by tissue, understand the role of either TORC1 or TORC2 in controlling growth of that particular tissue. And we eventually look at all the tissues, like a Manhattan Project. We put all this together and learn how whole body growth is regulated. So to do that, we had to make a mouse where we could conditionally knock out one of the two complexes in a tissue of choice. So we made floxed alleles, which allowed us to knock out the raptor, the gene encoding raptor, which is an essential and specific subunit of TORC1, or a floxed allele of the Richter gene, and Richter protein being a specific and essential subunit of TOR complex 2. So we did this so we could knock out either TORC1 or TORC2. Knocking out both would be too severe. Knocking out either one individually in any tissue of choice. And then the question is what tissues to look at. And we decided to focus on the metabolic tissues. And the reason for that is that these are the tissues, tissues shown on this slide. These are the tissues which are most sensitive to nutrients, most sensitive to insulin, and also control energy metabolism in the body. And these are precisely the three inputs which activate the TOR signaling pathway, or network. So we thought that TOR would have a particularly interesting or important role in these tissues. So we did that, and let me just give you a taste of what we've done. We've done a number of tissues, and we've sent these mice to collaborators, so many tissues have been done by now. But here's what we found in when we knocked out TOR complex 1 in fat, in adipose tissue. We found this mouse was resistant to diet-induced obesity. It could eat whatever it wants and never gets obese. Here's the wild-type mouse on the high-fat diet. This mouse is in fact metabolically super healthy. It has low insulin levels, low cholesterol levels, which then gets at this issue I mentioned before about rapamycin extending lifespan. In the rapamycin experiment, you give rapamycin to the mice, but this goes into all tissues. And then that begs the question, is there a particular tissue in which reducing TOR activity would give you the beneficial effect of extending lifespan? And now this experiment suggests that reducing TOR complex 1 in adipose tissue might play an inordinately large role in controlling a lifespan. We haven't done the experiment to test that directly. I can't get anybody to set up the experiment because it takes a it takes too much altruism by a student in the lab to set up an experiment for someone who will then complete after they've left. So it's still an open question. In any event, so that's what we learned by knocking out TOR complex 1. In the case of TOR complex 2, we had made the unexpected observation the mice are actually larger. The knockout is actually larger. And this then led us to identify a new signaling axis between tissues which controlled levels, circulating levels of the insulin-like growth factor hormone insulin, which are promoting this enhanced growth. And we think the logic of this is that TOR complex 2 itself responds to IGF and insulin. And if you knock out TOR complex 2, you knock out this negative control of IGF by liver or of insulin by the pancreas, such that these levels get very high and drive this growth. So this is a way of regulating circulating levels of insulin and IGF, such when these levels get too high, TOR is hyperactivated and then shuts down their synthesis. So very interesting observations have come out of these mice that we've published over the years. Now, while we're trying to understand the physiological role and the biochemical role of TOR, we've also been trying to understand TOR at the atomic level. This is the structure of mammalian TOR complex 1. It's a dimer of heterotrimers. So 2 copies of TOR, this is one, this is the other. 2 copies of Raptor, here's one, here's the other. And then the third protein, list 8 here and list 8 here. As it's a dimer, it has 2 catalytic sites. There's a catalytic cleft here and here. So the catalytic site is in the bottom of this cleft. We also determined the structure of TOR complex 1 bound to its inhibitor, rapamycin FKBP. And this then gave us the answer why rapamycin alone, although it binds TOR, cannot inhibit TOR. It needs to bind along with FKBP. So keep your eye on this part up here and this part down here. Voila. So this is now FKBP protein, and it's being stuck to TOR by rapamycin. Rapamycin acts as a kind of a glue which sticks FKBP here and here. And the effect of that is it creates a lid over the catalytic site, which sterically hinders access of substrates to the catalytic site. So that then is why you need FKBP, because rapamycin alone is simply not bulky enough to mask the catalytic site. Very unusual mechanism of inhibition of a kinase, but that's the way it works. Now, it took us about 10 years to determine the structure of mammalian TOR complex 1. And there are a couple reasons for that. One is we didn't have cryo-EM in the early days, but there's another reason. It's a very large complex that's shown here. And if you can't really get a feel for how big it is, but if we now stand it up, and then as a reference point, something the ribosome. So TOR is about as big, the complex is about as big as the ribosome. It's an extremely large kinase. It's much larger than I thought it would be. But what should catch your attention is the fact that this structure here is a very dense structure as opposed to the structure of TOR complex 1. And I think that illustrates how much is yet to be learned about the TOR complexes. I think once we know the structure of the TOR complex is bound to all its regulatory proteins, the structure of TOR will look like the structure of this. And these structures are being determined by us and by others, and some are actually have come out recently in publication. While we determine the structure of mammalian TOR complex 1, we also determine the structure of mammalian TOR complex 2. And there we found that TOR complex 2 is also a dimer, in this case of heterotetramers. TOR itself has the same structure in both complexes. It has different subunits which bind in the same regions of TOR, so adorn TOR by binding the same region to ultimately create a functionally and structurally distinct kinase. I think the most interesting thing we learned about the structure of TOR complex 2 was the answer to why TOR complex 2, as I told you before, cannot be inhibited by rapamycin, whereas TOR complex 1 can. So again, here's FKBP bound to TOR complex 1. No FKBP rapamycin can bind to TOR complex 1 because this is something we've known since 2004. And now we know that this subunit of TOR complex 2, Richter, masks the FKBP rapamycin binding site. So FKBP rapamycin cannot bind TORC2 at all. So that solved the recent resolution of a long-standing mystery. Okay, I've given you a rather long introduction, historical introduction to mTOR signaling. Now let me get to the recent story which we're so excited about, and that answers the question, how does obesity or diet cause diabetes? And as many of you know, this is a very timely problem because there is a very high and increasing prevalence in diabetes worldwide. So the darker countries are the ones where diabetes prevalence is particularly high. And as you can see from this, this diabetes epidemic is particularly severe in the Arab world. And we can focus in on the Arab world. Here in Saudi Arabia, the prevalence of diabetes is over 30%, which is really remarkable. It's indeed shocking. Here in the United Arab Emirates, it's 25%. So this is a serious problem, and particularly serious in this part of the world, which makes the story I'm about to tell you particularly relevant here in this part of the world. So, as you know, diabetes is a disease of blood glucose regulation. So let me, in rather simple terms, explain what this is all about. The body goes to great lengths to control the levels of glucose in the blood, and diabetes is a disease where this does not occur properly. So glucose is important. It's our main energy source, but glucose levels cannot get high. The body brings them back down when they get high, and this is seen over the course of a day. If we have meals, when we have a meal, our blood glucose level spikes, but then mechanisms exist to bring this back down to acceptable levels. And what happens in the case of diabetes, when we have a meal, our glucose levels again spike, but the levels of glucose do not come down to these healthy, normal levels. And this is a problem because this high level of glucose causes what's called glucotoxicity, which is the non-enzymatic glycation of proteins. And this then is what leads to all the diabetes-associated morbidities such as stroke, kidney failure, and vision loss. So the body has to control blood glucose. Too much is bad, too little is bad. It has to keep it in a very narrow range. So how does the body actually do that? Well, when we have a meal and blood glucose levels go up, this stimulates the beta cells of the pancreas to start secreting insulin. Insulin then goes in the bloodstream and acts on these peripheral tissues and stimulates them to take up the glucose from the bloodstream. So the insulin binds insulin receptors on all these tissues, which are on the surface of all these tissues. That then stimulates glucose transporters in these different tissues, and they then serve as a kind of sponge which soak the glucose out of the blood. Now, in the case of type 2 diabetes, what happens is insulin is produced, but these cells are no longer responsive to the insulin. And as a result, these cells can no longer take up the glucose. The glucose transporters are not stimulated. In addition, we know that diet is a risk factor, particularly obesity is a risk factor for type 2 diabetes. So somehow obesity can promote this loss of responsiveness of these peripheral tissues to insulin. So the question we set out to answer was, how does a high-fat diet lead, well, first of all, how do these tissues lose the responsiveness to insulin, and how does a high-fat diet promote this loss of responsiveness to insulin, or lead to this insulin resistance? So we approached this problem in mice. So this is what normally happens in mice. Glucose is taken up into the cell, and here we're focusing on adipose tissue, so fat cells. When there's high circulating blood glucose, insulin binds the insulin receptor on these cells. That stimulates through the mTOR pathway. In this case, mTOR2 stimulates activation of the glucose transporter called GLUT4. That causes these cells to take up glucose from the blood, metabolize the glucose, and converts the glucose to lipid. And this is the way this fulfills the function of the adipose cell, or the fat cell, to store the energy inherent in glucose in the form of lipid. Now, what happens in the case of an obese mouse or a diabetic mouse? We assume that the cells would no longer respond to the insulin. In other words, the insulin signaling pathway or TOR activity would not be stimulated by insulin. As a result, you would not stimulate glucose transport into the adipocyte, and therefore glucose levels would stay high outside. So to look at this, what we did was to look at the insulin signaling pathway in the adipose tissue of obese and lean mice. And this was our first surprise. We found that, so what we're looking at here are proteins which are phosphorylated by the insulin signaling pathway. And as you can see that there's no difference between this activity of the insulin pathway in a lean mouse versus an obese mouse, despite the fact that these obese mice have lowered glucose uptake, so higher blood glucose. So this was a complete surprise to us. Actually, David James and Sydney had published this earlier, but I never believed it. It was so heretical and I'm not so sure he believed it either, but he turned out to be absolutely right. So I take my hat off to David James. But this then creates the question, if it's not loss of insulin signaling that is responsible for the loss of insulin responsiveness, where is the defect? So this is what we saw, much unexpectedly, that the insulin signaling pathway is perfectly normal, yet glucose is not being taken up by these cells. So where's the defect? So to understand where the defect might be, we isolated adipose tissue from these two mice. The lean and obese mice, we did proteomics and looked for proteins which change in one versus the other. And this is, and what we found was a protein called hexokinase 2 was lost in the adipose tissue of the obese mouse. So what is hexokinase 2? Hexokinase 2 is the enzyme which mediates the first step in the metabolism of glucose, and the first step in the glycolytic pathway. It phosphorylates glucose and converts it to glucose 6-phosphate. And this caught our attention because we knew that if glucose is not phosphorylated, it will simply be transported back out of the cell. But once it's phosphorylated, now this large charged phosphate group is added, it can no longer be transported back out of the cell. It gets trapped in the cell and then further metabolized, converted to lipid. So if you don't have hexokinase 2, the adipose tissue cannot concentrate the glucose, cannot be this sponge which soaks up the glucose from the bloodstream. At least that was the theory. So we decided to focus on hexokinase 2. It could, in theory, explain why mice on a high-fat diet which have lost hexokinase 2 have high blood glucose. This simply confirms by blotting what we've seen by mass spec. We also assayed hexokinase activity not just looked at a band on a gel. We saw that indeed there was lower hexokinase activity in the adipose tissue of the obese mice versus the lean mice. And we also got human specimens, and there was also a detectable reduction in hexokinase activity in the obese tissue of obese patients versus healthy lean individuals. We couldn't blot for the humans because the antibody which they're available do not recognize human hexokinase. So whatever the mechanism here is, it seems to be conserved from mice to humans. So what we find about this problem in mice, we think applies to human. So is this effect specific to adipose tissue? Hexokinase 2 is expressed only in metabolic tissues. So in adipose tissue and in muscle. And we find that it is indeed specific to fat. Here's loss of hexokinase 2 in the obese mouse, in the fat tissue of the obese mouse versus the lean mouse. If we look in muscle of the obese, we still have hexokinase 2, much like in the lean. So it is specific to this one tissue. Is this a reversible effect? What if we, so here are the mice on a normal diet, hexokinase 2 is expressed at normal levels. In this case, they're put on a high-fat diet for 2 weeks, hexokinase levels go down. If we then put them back on a normal diet from the high-fat diet, hexokinase 2 returns. And if we assay blood glucose in these mice versus these mice versus these mice, blood glucose levels always correlate with levels of hexokinase 2. So when there's little hexokinase 2, there's high blood glucose. So this correlation suggests to us that there's a causal relationship between loss of hexokinase 2 and high blood glucose. Of course, it's only correlation, but we think it suggests there's a causal effect, and I'll come back to this later. So to see if loss of hexokinase 2 could account for high blood glucose or loss of glucose transport, we made a stable knockdown adipocyte cell line, and that's shown over here. Loss of knockdown of hexokinase 2, we lose hexokinase 2, and now we look at insulin-induced glucose transport, and indeed these cells which are insulin-induced glucose concentration in these cells. We find that cells which are lacking hexokinase 2 have a defect in concentrating glucose, much as the theory predicted. So this is where we were. We thought we'd had a pretty important observation, but it seemed too simple. It actually got me very nervous. I was rather skeptical. People have been trying to understand this problem for 30 years. It could not be this simple. Why did, there has to be something else. Why didn't people see this before? Is there some other orthogonal line of evidence which would support hexokinase 2 as the event which triggers high blood glucose and eventually diabetes? And here we looked around, and then here I have to make a rather abrupt transition and tell you about cavefish. So these are very interesting fish. They're from Mexico, and normally they live in rivers on the surface. But about 1 million years ago, these rivers flooded and ran into caves. And this carried fish into these caves. And since 1 million years, the fish which flowed into these caves had been evolving. And they did it in many different caves, so they've all been evolving separately in all these different caves in Mexico. And here's one of these so-called cavefish. And one way they evolved, you can see here, they've lost their eyes. Because they live in complete darkness, they have no use for eyes. But they have also acquired another trait, which allows them to adapt not to the darkness, but to the low nutrient conditions in caves. So these caves have nutrients only during the rainy season, so let's say 3 months out of the year, when nutrients flow in from the outside. They eat, and they have to essentially survive for 9 months with little to no nutrients. Absolutely fascinating organism. And this is something that caught the attention of Nick Rohner at the Storrs Institute in the United States. And he found that the adaptation to low nutrient conditions was high blood glucose. Now, why this should be an adaptation to low nutrient conditions is not really known. I can provide some speculation later if you like. But the fact is, all these different cave fish, and these are all different caves, so these are independent events, they all evolved to have high blood glucose. And how they can tolerate this high blood glucose is also a fascinating question for which we don't have the answer. So Nick Rohner assumed that the defect in these fish, which led to the high blood glucose, was a defect in insulin signaling, much like we assumed for our mice. So he sequenced the insulin receptor gene in all these fish, and he found that in 2 of the fish there was indeed a mutation, which would explain the high blood glucose. But in 1 fish, this so-called Molino fish, had normal insulin receptor, normal insulin signaling. And this caught our attention because this fish phenocopies our mice. Normal insulin signaling, high blood glucose. So we wondered, what could the defect be in this fish? Could it be a defect in hexokinase 2, like we detected in our mice? So we contacted Nick Rohner and said, sequence the hexokinase 2 gene in this fish. So he did that, and hexokinase 2 is a very conserved protein. It's conserved throughout all vertebrates. Here is a sequence of this protein in all these different organisms. He sequenced the surface fish, the parental surface fish, the 2 cave fish, also known as blind fish, the 2 cave fish that have a mutation in the insulin receptor gene, and they all were the same as all others. But when he sequenced the Molino fish, he found a mutation, hexokinase 2 gene. It converted this highly conserved arginine to histidine. So if this is in fact accounting for the hyperglycemia of these fish, in other words, is this a genetically fixed version of what we're seeing as a physiological response in mice? This would have to be a loss of function mutation because the mice, of course, are losing expression. That's a loss of function. So we model the effect of this mutation on the structure of hexokinase 2. We find that this arginine forms a key salt bridge, which stabilizes the protein. So if you mutate it, this arginine, you would lose the salt bridge and destabilize the protein, which should, in theory, give you less hexokinase activity. We tested this directly by expressing, in this case, rat hexokinase with this cavefish mutation, the arginine to histidine mutation. Introduce this along with wild type as a control into these cells, and then assayed total hexokinase activity. And we find that indeed this point mutation reduced hexokinase activity. So it appears to be a loss of function mutation. But we want to do one more experiment. Most people humanize mice. We decided to cavefishize our mouse. So using CRISPR, we knocked in this cavefish mutation into the mouse. And of course, the question is, does this mouse have high blood glucose? We have some indication it does. Hexokinase is an essential mutation, an essential function. So these mice do not do very well, so we're not absolutely sure about this, but there's some indication that if we have this point mutation, we have high blood glucose. So everything was looking good, but there was a problem with this mutation, or this experiment where we knocked in this mutation in the mouse genome, and that is when you do a CRISPR knock-in, you knock it into the whole mouse, all tissues. And in our case, we're on a high-fat diet, we're losing hexokinase 2 only in adipose tissue. So this is not really a very good experiment. So we wanted to knock out hexokinase 2 only in adipose tissue. So we obtained a flox allele of hexokinase 2. We knocked it out with a adipose-specific Cree driver. We could do this. And then we did a glucose tolerance test. Are these mice, which are now lacking hexokinase 2 only in adipose tissue, have high blood glucose? And indeed they do. So this experiment, the way this is done, is to inject glucose in these mice and see how long it takes these mice to bring their blood glucose levels back down. The mice which have this adipose-specific hexokinase 2 knockout are very weak at controlling their blood glucose levels. So this was great. The problem here is that this effect is too good, in fact, because fat takes up only 5% of our blood glucose. Most of insulin-induced glucose uptake is done by the muscle, the vast majority. And this effect here is too good to be explained by loss of uptake of only 5% of blood glucose. So something else has to be going on here. And here we then started considering the non-tissue autonomous functions of adipose tissue. So it's known that adipose tissue, when you have a high blood glucose, signals somehow to the liver to prevent the liver from producing glucose. So if you have low glucose, for example, you've not eaten for quite some time, the body compensates by making glucose mainly for the brain. And this is sensed somehow through adipose tissue, which then, when there's low blood glucose, it relieves this negative regulation of glucose production by the liver. So we reason that perhaps when we lose hexokinase 2 in adipose tissue, we are losing this negative regulation, and this is having the additional effect of leading to glucose production, which leads to this unexpectedly high levels of blood glucose in the hexokinase 2, adipose-specific hexokinase 2 mutation. So we wanted to test whether indeed there is glucose production going on in the liver upon knockout of hexokinase 2 in the fat. And the way you do that is now inject pyruvate into these mice, and if gluconeogenesis is going on, the liver makes glucose from pyruvate, a process called gluconeogenesis. So if there's gluconeogenesis going on, this pyruvate should be converted to glucose, and this is indeed what we see. There's more glucose production in the knockout versus the control. And to see that it's indeed via the liver, you can look at expression of the gluconeogenic genes in the liver, and we find that these are upregulated in the adipose-specific hexokinase 2 mutant compared to the control. So we think this is also contributing to this high blood glucose, this non-tissue autonomous effect of adipose tissue. So we can get back to the problem now of how does a high-fat diet lead to loss of hexokinase 2. We looked at this, and we found that here again is hexokinase 2 levels after 1 week, 2 weeks, 4 weeks on a high-fat diet. We lose it rather rapidly. The protein disappears, but the mRNA is unchanged. So it's not a transcriptional effect, it's a post-translational effect, which we now have to investigate in more detail. So we are in fact doing this. We have to understand how high-fat diet leads to loss of translation, and we know it's a translation elongation effect of hexokinase 2 mRNA, and we also have to now understand this non-tissue autonomous effect of adipose tissue signaling to the liver to dampen glucose production. But let me just close now by summarizing everything that I've told you on this second story. So we think in a normal, healthy individual, the adipose is taking up glucose in response to insulin signaling through the mTOR pathway. But in the case of a high-fat diet, we quickly lose hexokinase 2, which prevents the adipose from now concentrating the glucose from the bloodstream. This accounts for some of the glucose that's in the blood, but only a very small amount of it. I think the most of it is due to this concomitant loss of negative regulation of glucose production by the liver, such that there's a double defect here. This is now pumping out more glucose while the adipose tissue cannot take up glucose, and this then leads to these exceptionally high levels of blood glucose in these mice. Of course, we'd like to eventually not only understand this step here and how the high-fat diet leads to loss of hexokinase 2, we'd of course eventually like to understand, use this information to maybe develop a therapy which would prevent the high-fat diet from turning down hexokinase 2 to somehow treat hyperglycemia and therefore eventually prevent diabetes from developing. Okay, let me close by giving credit to the people who did the recent work I told you about. This is all the work of an outstanding fellow in the lab by the name of Mitsugo Shimobayashi, working with one of my long-term technicians, and other postdoc Sunil Shetty, and a student, Irina Frey. They alone did all this work on high-fat diet causing hyperglycemia. The structural work, which was also recent, was done in collaboration with my colleague at the Biozentrum, Tim Meyer, and also with Nenad Ban at the ETH in Zurich. And then the cavefish part of the story was done in collaboration with Nick Rohner at the Stowers Institute, United States. Okay, thank you for your attention. I'm happy to take questions if you have any.